So last 2 weeks we were discussing about the very basics because the objective of this course for you to explore any genome, your genome or like probably a like COVID genome or like the COVID virus genome or like any genome. So in order to do that, we have to understand the basics. So far, we covered almost like all the basics, the bare necessity. So so you have a very good understanding on how to interpret things in the coming sessions. So today we are jumping into the one of the most interesting aspects of the exploring genome, that is mapping genomes. And I'm pretty sure the moment you hear about the mapping, so I mean, I'm pretty sure you visualize a map, like if you play games, especially if you're a fan of the Bethesda game, the Elder Scrolls or the Fallout, the maps. You can't live without maps. So you go explore and like an open world games. So when it comes to our genomes, it is the same thing. So we have like a lot of open endings, the best RPG made ever. So you can play around, explore, and find things. So exactly that is what exactly we are going to do during this course. So when you look at a map, it's usually positions based on interesting features and other important landmarks. So that's how we know, okay, so this is that, and this is how we move forward. And when it comes to a genome map, can you guys let me know, what are the landmarks and features you expect in a genome map? So you can use the chat window and quickly type one or two things you might see in a genome map, because I would like this class to be as interactive as possible. Okay, so go ahead and maybe I'll wait as usual. I'll wait for like two answers, and then we can move forward. So the question is, what are the landmarks and features you might find in a genome map? Okay, so thank you, Asanti. So you mentioned the two very important things. So one is genes, of course, and also you mentioned gene markers. So we are going to talk about all of these aspects yeah, very soon. So at least now I know you. Most of you have some understanding about genome maps. Okay, moving forward, as you said, the some of the key landmarks includes genes and other distinctive DNA sequences. Because when we take a genome, genes is remember last time we talked about the human genome. So it's like 50%, you have repetitive elements, and then the other 50 is what we were the scientists were mostly interested in earlier, but now things change. So when it comes to a genome map, so we have to have some kind of a strategy or approach, right, in order to make a genome map. So there are two very common approaches followed by scientists, and these two approaches includes genetic mapping, and then the second one is physical mapping. And so these are the two strategies followed by scientists for a long period of time. You will see one approach is very old, and it goes back to a long period of time, hundreds of years. And then the other one is relatively new. Relatively, it's not brand new, but relatively new. So we will start with genetic mapping. And when it comes to the genetic mapping, uh, it's let's watch this video first and then see.
Welcome to Moo Moo
Can you
Math
hear
and
now?
Science. In this video, I'd like to talk about
Yes, sir. Now
hemophilia,
it's okay.
the
Awesome.
royal
Okay.
family, and an X-linked disease. Hemophilia acquired the name the royal disease due to the high number of descendants of Queen Victoria afflicted by it. The first instance of hemophilia in the British royal family occurred with the birth of Prince Leopold. Hemophilia is an X-linked recessive disorder. The blood of a hemophiliac lacks the ability to clot due to the fact that one or more of the plasma proteins required to form a clot is absent. A recessive gene is carried on the female chromosome X. Males possess an XY chromosome, and females have an XX. Since females have two X chromosomes, they are more often carriers and not have the disease. If you look at a Punnett square, you can see that each child of a carrier will have a 50% chance of inheriting their mother's mutation, of being a hemophiliac son or a carrier daughter. The daughter of a male hemophiliac will always inherit his mutation, while the son cannot ever inherit it if the mother is not a carrier nor has the disease. A female will only be affected with hemophilia in the rare circumstance that she inherits a mutated X chromosomes from both a hemophiliac father and a carrier mother. No case of this inheritance is known among Queen Victoria's descendants. Because a female can be a carrier of the disease, it can spread, as was the case with the royal families. Princess Alice and Beatrice, both of whom were carriers, spread the disease to other royal families. Queen Victoria married Prince Louis and had 5 daughters, 2 of which were carriers. Her daughter Alice had a daughter Irene who was a carrier and married into the Prussian family and had 2 sons with the disease. Alice also had a 4th daughter, Alex, who spread the disease to the Romanov family with her marriage to Nicholas II. Queen Victoria's daughter Beatrice had a daughter, Victoria Eugene, who was a carrier and married Alfonso and spread the disease to the royal house of Spain. So there we go, hemophilia, the royal family, and an X-linked disease. Thanks for watching, and Moo Moo Math uploads a new math and science video every day. Please.
Okay, so it's one of the very interesting the stories where we use one of the approaches of genetic mapping to map the history of the disease, and that's why it's used as a very classical example. So therefore, next time if you're getting married to a royal family or like a big political family, so be little careful. So you never know. Probably they're because. Uh, Sometimes they can carry this genetic disease. The only thing is if it happens in a royal family, everyone will know about it, right? And then it's easy to trace. But if you are common people like us, so no one really going to do a pedigree analysis and see whether how this whatever the trait carried from one generation to another. Anyway, moving forward. So in when it comes to genetic mapping, we are mostly doing the linkage analysis. So here, in order to do the linkage analysis, we base it on genetic techniques, and to do that, we have to do planned breeding experiments. Okay, so now probably, I mean, this is really cool because I mean, so most of our colleagues in our department, they do this kind of planned breeding experiment. But now imagine you want to do a planned breeding experiment with humans. Can you do it? Like it's going to be impossible, right? Because here we can easily get one plant and another plant and then do the pollination, and then we can get the fertilization and have the progeny. What will happen if you go and grab one human and another human, and then you breed and do experiment? So you will be in jail. So therefore when it comes to plants, plant breeding experiments are easy to carry out, and it's same for other animal models like mouse. So if you take the danio, the zebrafish, or whatever other models, but not with humans. So when it comes to humans, what we do is we basically look at the family histories, as you already witnessed, and we do these pedigree diagrams and then try to trace back these kind of mostly the start diseases, but you can do it for like other characters as well. So this is so you guys can find out who this person is, because that earlier video already explained this about this queen. Okay, try to find her name.
Welcome to Moo Moo Math and
Sorry.
Science. In this video, I'd like to talk.
Sorry about that. And so if you, I mean, I included a very classic paper explaining the pedigree of hemophilia in royal family. And so I already put it into my Rakuten Drive. So you can download and read this paper. It's a very short paper, but I'm pretty sure you guys are going to enjoy the paper talking about this royal disease. Okay, so the first approach, as I mentioned earlier, it's genetic mapping, and then the alternative approach is to use the physical mapping. So if you ask yourself, what is the difference between this genetic mapping versus physical mapping, as name implies? So in physical mapping, what we do is we use molecular biology techniques to examine DNA molecules directly. So doing that, we can identify the positions and specific sequence features, including the genes. So therefore, this is more new approach compared to the genetic mapping. Okay, so these are the two major the approaches we use. Today we are going to talk about genetic mapping, and next week we are going to talk about physical mapping. And so now you're thinking, okay, so we are talking about genetic mapping, physical mapping, and then like the I'm pretty sure you have this question, why we need to do these kind of a mapping? Okay, because it takes time, and like sometimes like some mapping experiments, so you will go like crazy, like when it comes to analyzing data and like to try to find missing links. So this is basically we are going beyond Mendel, and all of you remember, right? George Mendel, and and his breeding experiment. So he and the scientists put the foundation for mapping genomes, and so as a result, now we have the ability to build on that and make a very detailed map. And when you have a very detailed map, and this can be essential prerequisites for the assembly of correct sequence of a genome. Okay, so we use map as a blueprint in order to assemble our genome. Okay, but uh, so times have changed, we are moving very very fast. uh, so therefore we have more advanced approaches, and then we our dependence on genetic map can be less important, or less compared to earlier days. Okay, so those days the major bottleneck was the sequencing. And can one of you tell me what is the normal length of a Sanger sequencing output? So I'm not talking about all the bases you receive from whatever the sequencing provider, but what is the usable length you get from Sanger sequencing? Can you guys quickly type based on your experience? My question is what is the usable length you get once you send a sample for Sanger sequencing using your own primer? How many of you, I mean, so probably you can say yes if you done any Saga sequencing experiment earlier, or recently, or whatever at one point of your life. How many of you conducted you can type yes if you send samples for Saga sequencing and obtain results at one point of your the undergrad or postgrad. Nobody did Sanger sequencing. That's surprising. I don't think I'm going to believe that. Nobody? Don't worry, I'm not going to ask a follow-up question if you say yes. That I promise. I guess next time I will make a polls or quizzes for the lecture. So therefore, like I can get easy answers. Okay, so we have one conducted. So usually, what we get is around 750 base pairs, and so Sri Lanka, like if when we send samples to sometimes gene tech, except for gene labs, so like sometimes we only get 400 base pairs, 750 if you're lucky. So therefore, like this is going to be a issue because when you sequence like a large molecule, so you have to like basically overlap these sequences in order to get the full sequence. And if you can remember, your genome is 3.1 billion uh, bases, and it's spread across 23 chromosome pairs, right? So now imagine assembling your genome. I mean, this is how this initially did the human genome map. We will talk about that approach as well. So therefore, it is not an easy thing. Those days, that was, I mean, this is the bottleneck scientists are facing. Okay, so therefore they wanted to come up with a different approach. And I don't know how many of you had the chance to use a shotgun. I have, so when I was abroad, I had the chance to use a shotgun, and you know what shotgun does, right? So because we are going to talk about like a shotgun base thing for the people use in order to do the sequencing. Okay, so this is what we call as I mean not the actual shotgun, the approach scientists used in order to reduce the issues associated with the short sequencing rate. So they are using this method called shotgun method for the sequence assembly. What they did is basically when you have your DNA, you basically break it into small fragments, and then each fragment was sequenced, and then they did the assembly based on the sequence similarity uh, between this because you get overlaps, and based on the overlaps, so you can assemble your genome. Okay, and so that's yeah, give me 1 second. Right, okay, now it's set. Okay, so you can see here, so you have a common sequence, and then so with that, because when we make fragments, so these fragments are most of the time overlapping. So therefore, we have the chance to align them. I think those who work with Sanger sequencing, you do sequencing from this side to this side and this side to this side, and then you overlap and get your consensus sequence for your experiment. So that's how you do it. So we do same with the shotgun sequencing. Okay, so but as I mentioned earlier, so there are issues, especially if you're working with large genomes. Uh, Imagine it might not be possible to obtain sufficient short sequences to produce continuous or contiguous DNA sequence for the entire genome, because our genomes are huge. Okay, so they, but I mean, therefore, I mean, whatever the results you're getting is actually a genome sequence made up of many short segmented uh, sequences, and they're separated by gaps. Okay, and then if there are gaps, that means you're only coming up with a part of genome. Okay, and then by chance it's not covered by the sequence that have been obtained from your shotgun sequencing. Okay, so this is the main issue. associated with shotgun sequencing when it comes to the genome assembly. But this still can be fixed. That's how they, came up with initial genomes. And the solution was to identify these different segments based on your genome map. Okay, so because remember I told you we are going to talk about the why we use genome map. So now you have one reason, because you have small sequences here and there are some gaps. But if you have that sequence along with the genetic map, so that means now you have a kind of blueprint or footprint to assemble your genome even though there are gaps. Okay, so how you do it? We basically anchor our segments onto the map, and then thereby we can obtain the correct genome sequence. Okay, so therefore we are basically reducing the amount of issues caused by the gaps, something like this. If you look at this diagram, you can see you have your DNA fragments, and then you do your shotgun sequencing. So now you get like different sequence fragments with like whatever your markers or like the footprints, in this case genes. And now you have B, C, E, and F, G, but you are missing A and D. So, but with the help of genome map, you can basically map it to the that whatever the foundation or the blueprint, and then move forward. Okay, so this is how the initial genomes were sequenced. Okay, so but that's not the only problem. You had a sequencing bottleneck, and then you found some solution in order to overcome that. And then the second biggest issue. So this is something we discussed last week as well, because when we talk about the human genome, and same for like many plant genomes as well, so they have a large number of repetitive elements. Okay, so human genome, remember, it's like 50%. Okay, so this repetitive sequencing, they are going to cause issues, because if you look at this image, you can see, so you have your DNA, you have two copies of repeated DNA sequences, and when we fragment it, okay, so they are like identical. So that means the some parts will like broken into two, and then you will see there's a sequence complementarity. So that means instead of this one pairing with this, so when you assemble, you pair this one with this. So now you have a completely different setup. And because this repetitive sequence, they are located in a way, so they are like their length can be like an in between region as well. The kilobases is in length. Okay, and then like as I mentioned earlier, they repeated in many places. And then when you try to assemble, what is going to happen? So because of this so-called sequence similarity between repeated element, so you will have a wrong assembly. Okay, and if you have the genome map, again this can be overcome. Okay, okay, so this is basically like even if you connect these two, so you're basically separating them in different they're putting in different places. Okay, so the solution, as I mentioned earlier, you can use genome map as a blueprint to avoid this issue. Okay, so because if you think about your sequence, the features, and if it matches the genome map, then you can put it correctly. And if they are not matching, then you have to revise your assembly, because you know the genetic map. So based on that, so they are not fitting. So therefore, like you have to do something about it. Maybe another round of sequencing or a different approach in order to overcome these issues. So here you can see the if it's an ideal situation, you have to have like a very nice organization of your assembly. But here you can see it's not the case because of the repetitive elements. Okay, but now we know that our technology It advances a lot. Okay, so not only the sequencing technologies, but also the computational algorithms. Because now if I give you a set of sequence, so you just use like a very very simple program and then assemble. But good old days, remember, people they had to basic manually align sequences. You put one set of sequences into like let's say Notepad or Word, and then the reverse sequencing onto a separate part, and then you try to match them together. That's how people did it like in a good old days. Now we just drag two sequences into whatever our program like Genius or Snapgene or whatever the program you use, and then just one click, and then you can basically overlap them and get the consensus sequencing. So therefore, and also they have the ability to detect repetitive elements, and sometimes even you see when we do the genome assembly, you sometimes mask those the repetitive elements in order to improve your the downstream application of genome genome maps or like in genome assembly. Okay, so the end of the day, what is happening is the now the dependency on the genetic maps is getting less and less important. But you should know you because there are some uses. And when you think about the prokaryotes, it's piece of cake, right? So the bacterial genome you can assemble within a. So when I do some practicals for like other postgraduate programs, I give my students their own genome to work with, and probably I will give you guys as well. So what you do basically get it's very easy to assemble a prokaryotic genome with these tools. But if you work with crops like uh, especially barley and bread wheat, we are going to discuss in a bit. They have huge genomes. So when it comes to huge genomes, it's not very it's not a better process. So it's going to be very difficult, because if you take barley as an example, it has a genome of 5,100 megabases. If you compare it with your own genome, that is only 31,000 megabases. Okay, and this 5,100, 80% is repetitive DNA. Now can you imagine only 20 is non-repetitive DNA. So that means it's a nightmare to assemble it, because if you have so many repeats, so it's you don't know how to put them together. And if you take another example, this is the barley. If you take another example, that is like bread wheat, the one you use every day. So that plant contains three genomes. Okay, it's called A, B, and D, and each genome contains 5,500 MB. So that means if you take the bread wheat as like as a whole, its genome is 16,500 megabases. So yours is only 3,000. So that means at least five times bigger than the human genome, and it also contains around 80% of repetitive elements. So these kind of a cases, you have no option but to rely on the genetic map. So I have a small homework for you all, and you guys need to read about the barley genome project and wheat genome project. So you will see there's a lot of success, and you have to have an idea. So my advice is go and find the paper talking about the release of barley genome and bread wheat genome, and then see what really happened and how they did it. Because please remember, when it comes to my exams, you will not get direct questions. Maybe well, sometimes kind of well, let's say out of sympathy, I put a couple of direct questions, but very rarely. But all the other questions, I want you to think and synthesize your answer. So therefore, you need to be very well versed about things happening around. So therefore, if I say do some kind of homework, so please make sure to read those papers, because the exam is not going to be a straightforward exam. Uh, And so sometimes I even give take home exams because I'm not worried that like because it's like oh, even I am fine with open book exams because I am not going to ask things directly. And also when it comes to exams, please remember the if you use the chat gpt or the like llm models or like the bots, I can easily detect it because I have the access to a couple of tools, so therefore I can easily check. And also it's very simple because so most times I already checked like tools like chat gpt and like the if you take Gemini or like the many tools, their accuracy is like around 50 to 60% when it comes to scientific facts. So like some bots even like make a fake references. So therefore, it is very easy to detect when it comes to when it comes to correcting papers. Okay, just like small warning about. I mean, I'm not saying don't use these tools for your advantage, but not for the exams because it might bite you back if things go south. Okay, okay. So moving forward, um, so when it comes to the genome maps, as we already discussed, it's not just sequencing aids, uh, because uh, so they are more than that. Okay, so it's not just like A's, T's, C's, and G's, because so far, like in the good old days, so the question we were asking was, what does this gene do? Okay, but later on, we were thinking more reverse approach. What we were asking is like which gene is responsible for this characteristic or the trait, because both are equally important. Okay, so when it comes to the genome maps, they are especially important for the latter aspect. The asking question, which gene is responsible for this specific phenotype, character, or the trait. Okay, okay, so why? It's because the approach used initially involved the mapping the positions of genes being sought relative to other gene sequences, and these ones feature like whose location on the map is already known. Okay, so and this probably you think this is not important. Of course, this one part is also important because it helps us to identify like some major human diseases, starting from like cystic fibrosis to breast cancer. Okay, so therefore, like I know, like one of you are working on like the breast cancer related stuff, and cystic fibrosis. If you can remember, like it's a classic example because one nucleotide difference is going to cause this disease because there will be like an amino acid chain, and the result is you're going to get cystic fibrosis, and you know how bad that disease can be. And now with the gene editing, actually we can probably like one day we can correct this one as well. Okay, so and then like the similar methods are used in identifying genes and like the which are like possibly spread around the genome. The thing is, not all it's not always one to one, right? Because like some like having that specific defective gene is going to cause the disease, but some it's not simple as that. Like if you take most cancers, if you read papers you will see like so let's say like there's one paper talking about okay, so the we identified 13 possible loci associated with prostate cancer. So that means they are not like directly involved, but having that defective or like the mutation or the change can leads to the susceptibility. So that means like some are like very susceptible for like some disease, some are not. So that difference is governed by various loci or the genes involved in there. Okay, and there are more field applications, and these genetic maps are used to identify quantitative trait loci, especially those who are working with the breeding and like especially plant breeding. you can't live without the QTLs, especially like plant breeders. And these QTL are basically it can be like one gene. It can be multiple genes, and they are controlling like variable traits. As an example, sometimes, so if you take like let's say one QTL, that QTL can control how plant responds to water stress, like the lack of water or drought, and also probably like how quick their seedling is growing, which is called seedling vigor. And also, if you think about the animals, the meat productivity, like how much fat it's going to gain, and like how quick it's going to gain. So those things are possible. And also, when it comes to plant, whether they are like resistant to, let's say, different insects, or like pathogens. So those things, I mean the QTLs are like are like that. And you use genetic maps to identify these QTLs. And especially like this is very important when it comes to the commercially important crops, because we can then design our breeding programs in order to develop new varieties with improved agriculture properties. Okay, and I think one of you already mentioned about the markers. We are going to talk about several different types of markers today. And if you heard about the term MAS, the mark assisted selection, and that is also basically like it's a kind of a result of genetic mapping. Okay, and so therefore, so these kind of things we need genetic maps, and so therefore it's not archaic, so because you can still use it for like some aspects, but their importance is like getting less and less and less over the time. Okay, so next we are going to talk about the landmarks and features we discussed earlier. Maps and like markers, because you can't have a map without the markers. And if you think about like regular map, you have like rivers, roads, and building. So let's say I feel like eating a pizza, so like this is the closest pizza shop in near to my home, the Domino's one, and it's just like 6 minutes. So therefore, like if how I'm going to go there, so this will provide all the landmarks, and now I know because it's right next to the Apico Super Center, Katugastota. So therefore, like this is some. I mean, map is something like that, and so when it comes to the genetic maps, we talk about. I mean, like one of you mentioned about the markers. The question is like what markers can we use in this genetic landscape? How you find different footprints using the these specific signatures or the markers. Okay, so when it comes to the markers, genes were the very first markers to be used in this uh, genetic mapping uh, scenario. Okay, and you know who this is. So this is basically the fruit fly, and I am pretty sure you learn about the model species. Actually, I also introduced a model species and fruit fly. So because most Sri Lankans, when we say fruit fly, so they were thinking about this the fly with like like that basically damage the mangoes and other things, because sometimes when you open a mango you'll see like 100 the kind of caterpillars or larvae coming out. So and that fruit fly is not the fruit fly the fruit fly model or the one we use. So this is like during the April, like the New Year time, you see a lot of konduruvo, or like the flies. So those are the fruit flies or the Drosophila. The ideal model is the Drosophila melanogaster. And let's see, there's a small power failure. I hope I'm still connected. Yeah, no problem. Yeah. So the Sri Lankan one, that Kondurva is Drosophila some other species, but scientists use Drosophila melanogaster for the all these activities. Why I'm talking about fruit flies? Because fruit flies are like one of the very earliest model people use for the genetic mapping, and there they were using genes as markers. In order to use a gene as a genetic marker, so they have to have specific characteristics. Okay, so most importantly, these genes must exist at least in two forms, or what we call as alleles. Okay, and each allele should give a like a specific phenotype. So we can go back to Mendel's time. Remember, so Mendel was studying tall and short stems in pea plants. Okay, and those are like a two distinct characteristics controlled by two different alleles. Okay, so but there's a limitation, because if you want to begin with this kind of analysis, so you have to have the genes with specific phenotype, and that can be identified visually. Okay, so this is not simple as you think, because if you take an example, the early fruit fly maps, they showed the positions of genes in their genome based on different characters like body color, eye color, wing shape, and some things like that. And these ones you can easily understand, identify using low power microscope. Okay, so now you think, okay, so this is cool. Okay, so you see fruit fly with, let's say, some, let's say, red color. I mean, it's not, it's a hypothetical example. So you think, okay, so now you can link it to a specific allele, and then if you don't have that allele, so you have let's say yellow color. So now you have two distinct phenotypes. But are these only genes? You think every gene is going to show a phenotype? That's not the case, right? So therefore, the limitation is uh, so there are only few phenotypes that directly link with the different alleles. Okay, so therefore, the this is going to be a problem, but it was okay during the early days. But later on, the genetics they realized there were only a limited number of visual phenotypes. Okay, and then things got sometimes things get very complicated, because a single phenotype can be shown by various different genes, like more than one gene. Okay, as an example, in 1922, I mean, long time back, so they identified that over 50 genes mapped to the fruit fly chromosome, but out of that, 9 out of 50 are responsible for eye color. So if you look at this diagram, I don't know whether you can read, so there are various different eye colors. So therefore, it's not remember I told you, very distinct, and so you can't basically link one to one. Okay, so therefore, if you want to make maps more comprehensive, you have to find characters that are very distinctive and less complex than the visual characters. Okay, so can you guys think of an approach? You understood the issue, right? The issue is so not all what appears is related to a character. Okay, so probably you have very very nice friend, and you think that person is going to be nice sometimes. So they are nice until they get the opportunity. Then you realize, okay, so that trait is not directly linked to that. So there are other traits as well, because one sometimes one feature, there are multiple genes involved, so therefore it's not straightforward. So the what scientists thought, okay, so we need to think of a new approach. So what they found was that if you look at the biochemistry of your favorite organisms, so then you have a chance to find multiple alleles that are represented by different I mean, different biochemical compounds. Okay, so this is an example, as you can see here. Uh, so they use different markers. As an example, ADE2 And this phenotype, it requires adenine. So that means when you grow whatever your yeast, because here we are looking at the Saccharomyces cerevisiae or the brewer's yeast. So they grow only when adenine is present in that specific medium. That's a biochemical character, right? And then the same for the can one. And there are many different examples you can see. So basically, you're altering the medium in order to check that specific phenotypes, and you will be surprised sometimes. So they found there are more than 500 different variations of a character. So that means earlier he had like if it's the eye color, let's say it's like only two phenotypes, red or black. But now with the help of biochemistry, you can study even more characters, because now you have more chance of detecting these compounds. Okay, so there that means, so now you found an alternative for gene markers. Now you're using biochemical markers. Okay, so I'm going to give you, let's say, 5 minutes break. Now it's 5:52. We will meet at 5:57, and we are going to continue talking about different marker types and how the markers advance uh, with time. And now we have much better approaches than the original gene markers. So moving forward, I know sometimes this part can be like a little boring, but unfortunately we can't move forward without talking about this kind of what do you call old stuff. Okay, so because even though we don't really feel like it's so awesome, but you know, I have to do it. So sorry about that, but let's continue. And we are talking about the markers, gene markers and biochemical markers. So then, so sometimes we need even beyond that kind of a markers, because if you think about large genomes, and especially we talk about different vertebrates as well as the flowering parts, uh, so they are not they can't basically rely 100% on those the gene markers as well as the biochemical markers, and because we need a very detailed the map, otherwise it's it's less useful. um, the other thing I wanted to mention is, if you get a chance, go to the NCBI, because I'm pretty sure you all of you are very familiar with the NCBI. So go and take a look at the genome browse, JBrowse, where you can see all the tracks showing. Okay, so this is the gene, and these are the transcript likewise. And so there, if you zoom it out, zoom it out, right? Yeah, not zoom in, zoom out. You will see. So between genes, sometimes there are large gaps. Okay, some genes they are very closely associated, like in a kind of cluster, but some, so they are very mm, they're separated by sometimes thousands, sometimes even kilobases or the megabases. So I mean that can be that's a possibility, because if you if you check the plant genome, you will see the plant genes are relatively small compared to human genes. If you take your favorite human gene and you take a look at it, and maybe the adjacent ones, you will see some genes they expand megabases. And so therefore if you want to amplify that gene and study, you have to look at many Sanger reactions in order to cover the whole gene. But when it comes to plants, usually you can safely do maybe four reactions of the sequencing in order to obtain your gene, but not it's not the case for the animal models or in the animal genomes. So I mean that's one of the differences, because when it comes to the animals, so one important thing they have in order to become more diverse is alternative splicing. That is something we are not going to cover this course, but alternative splicing is very very cool thing, and it's one of the major ways. they basically make a diversity, because when it comes to plants, so they can multiply vegetatively. So that means you don't need to have like pollination, something like that. So as a result, so they can have like let's say multiple ploid levels and like a huge genomes. uh, so that's like one of the biggest advantage. So therefore, uh, um, if you take like any plants you find, so based on whatever the literature available right now, all plants underwent at least one duplication event. So that means, so they were like at least tetraploids during their evolution process. Okay, and so genes are like very far apart, and also you can see this is made worse because only a fraction of total number of genes exist in allelic forms. Okay, so if they are not in allelic forms, that means you can't conveniently distinguish them from each other. Okay, so therefore, your gene maps is not going to be very comprehensive. So that means we need a much better marker in this aspect. Okay, so now we went from gene to biochemical markers. Now we are going for another set of markers that you can use that helps you to have a very comprehensive map. So starting uh, when we start talking about these DNA markers, so as we already discussed, these DNA markers must have at least two alleles to be useful. And there are two very popular approaches. So starting with the RFLP, or popularly known as restriction fragment length polymorphism. And the second most popular one is the SSLPs, or simple sequence repeat polymorphism. Okay, so when you think about mammalian genomes, so they have around 10 into the power of, that's like 10 lakhs. That's money, right? 10. Uh, yeah. I'm thinking like we use lakhs for the like it's like 10 into the 5, so that means 100,000, right? 100,000 RFLPs in mammalian genome. And so we can use this one for our advantage. Now you're thinking, what is like restriction fragment length polymorphism? Because when you take our DNA and then when we take our restriction enzymes, if we cut your like the genome using a restriction enzyme, so you will have different, you will get different size particles. So then we can basically visualize them and uh, identify, okay, so this RFLP fragment is will act as a marker. So then based on that you can build your genetic map. So now like if you think about it, so this is a large number of RFLPs. So let's say you basically cut your genome with your favorite restriction enzymes. So you're going to get a lot of bands. As if we take an example, take eco R1 as example. It identify six nucleotide palindromic sequence GATTC. Okay, so if you think about the theoretical number of like if you think about like the theoretical distribution of ACTGs, so this eco R1 should cut approximately once every 46 bases. Okay, oh so and each fragment should be around 4096. And then, so this is going to yield around 750,000 fragments if you cut your genome with eco R1. Okay, so now imagine, okay, so you took your genome, you use eco R1, and now you have the digested, restriction digested, uh, the whatever the product. So now you're going to run a gel. Okay, so now imagine when you look at your gel, so what you're going to see. So there are 750,000 fragments of different size. Okay, what you're going to see is a big smear from like top to bottom, a smear. It's not going to provide any useful information, right? So why you see a smear? Because as I mentioned, because you have different size fragments, so therefore there is no use. Okay, so therefore we have to follow different approaches to visualize. So one, the very old approach people use is southern hybridization. So what you do is basically, if you're interested in the one specific polymorphic site, you are going to make a DNA probe. So this DNA probe, when I did southern hybridization to see we made a transgenic plant and we wanted to see how many copies of the newly inserted gene is there. So we were using DNA probes. That probe, you can color it using the chemiluminescent stain or radioactive. So my case, I was using P32 as the isotope as a marker. So basically, we label our bases with P32. It's radioactive, so you have to wear all this protective gear, and then you have this one what we call as a dosimeter, so it will measure how much radiation I was exposed to. Like every 6 months they check whether I was exposed to too much radiation. If it happens, I have to stop doing that kind of research. So you have to follow a lot of the restrictions in order to do that. And then what happened is, so once you run the gel, and then you basically use your probe to hybridize, and then you take the whatever you output and you basically keep it with the X-ray film. So what is going to happen is, the P32 is going to emit the radiation. So as a result, your X-ray, you are going to see a band uh, in the if you're lucky, the correct place. So then you can identify, okay, so this fraction is there, this is the mark, right? And then, but as I mentioned, it's not that easy as it seems. There's a lot of it's tedious, because most of the time we can only run 12 reactions at a time when we do a southern hybridization. But so imagine, if you have a gigantic genome and running 12 at a time, so then I mean, it will take forever for you to complete your, let's say, your project. So instead, now we use PCR. So what we do is, we know that, the based on some footprints, uh, okay, so this is located here, this is located there. So we use PCR primers to amplify that region with our whatever our known sequences, because in the middle we have no idea what it is. Our idea is to basically find this polymorphism and then the related marker. So we amplify this PCR product. Now instead of restriction digestion, the whole genome. So rather, now we basically cut the PCR products using our enzyme, and then we can easily visualize it in a gel. So this is how we visualize the RFLP markers. Okay. So the second type is SSLP, simple sequence length polymorphism. And so these are basically uh, an array of repeated sequences, and they show length variations. Okay, so that means, so when you have a one allele, so they can contain different number of repeat units. Okay, if you compare this one with RFLP, the SSLPs can be multi-allelic, because each SSLP, they have a different number of length variants. So this is a big advantage, because I don't know whether if you had an experience with SSR, remember, when you talk about codominant markers and dominant markers likewise. So then you can basically separate the codominants using these markers as well. And there are two types of SSLPs. One is mini satellites, or they are also known as VNTRs, or variable number of tandem repeats. And here, these ones, so usually the repeat tune is it up to 20 base pair, 25 base pairs in length. Compared to that, the microsatellites, or what we call as short tandem repeats, or STRs, so their repeats are shorter, usually 13 base pairs or less. Okay, and so you'll see. which one is ideal for like most of our scenarios in a bit. But before that, like if you want to have a like a visual idea about this, so you can see your allele 1 contains GA, and if you look at your allele 2, you can see there are more than there are two sets of additional repeats. So therefore, you can basically separate these ones using your SSLP markers. Okay, so now you're thinking, okay, so why it is best, micro or the mini? So let's see the battle of minions. And if you think about, like, if you ask those people who do this kind of studies, they will tell you, of course, we are going with microsatellites because microsatellites are more popular than mini satellites. The number one thing is, if you look at mini satellites, they are not spreaded evenly throughout the genome, and they have some kind of a bias. So they are usually located close to the telomeric regions or the ends of the chromosome. Okay, so that creates issues because let's say if you want to find middle of an island. Okay, so you live in Sri Lanka. No? Sri Lanka is an island. So we have lighthouses in like Pedro, and then the whatever the bottom one the Undar, then you have like lighthouses and probably like West and East as well. So you want to study the features of Candy. Can you use those lighthouses as a guide? Of course, no, right? So now imagine about the mini satellites. Mini satellites, they are located close to telomeres. So they are, that means towards the end. So therefore, um, using mini satellite is like trying to use a map of lighthouses to find your way around the middle of an island. So therefore, like you will get less or like zero success with mini satellite comparatively microsatellites. So they are more conveniently spaced throughout the genome. Okay, so the quickest way to type length polymorphism is through the PCR. But if you think about your PCR reactions, you can do it very fast and accurately if your sequence, the product of your sequence is less than 300 base pairs length. Okay, and if you're thinking why I'm talking about this, because most mini satellites, so they are longer than this because as I mentioned earlier, because they are located towards the end, so they are repeated units, they are relatively large and they seems to present in single arrays, not multiple arrays. And the result is going to be your PCR product is going to be several kilobases length. Okay, so that means it's not convenient compared to microsatellites. If you take the microsatellites, when we use them as a DNA markers, they uh, typically contains 10 to 30 repeats, and they are no longer than 6 base pairs in length. Okay, so that means it's more convenient or much amenable for the analysis in PCR. Okay, and if you think about our genome, there are more than 4 million microsatellites with repeat units of 2 to 6 base pairs. Okay, so that means so the microsatellites are going to provide like an ideal marker over the mini satellites when it comes to developing our genetic map. Okay, now probably you're thinking how we are going to type these short tandem repeats using PCR. So there are two ways. So one is the conventional way. You can use the first the PCR amplification followed by the agarose gel electrophoresis. But it will take some time because when you make gels, so you have to do it's more time consuming, but now we have much better option. So we have a capillary electrophoresis. So then basically you run your sample, the machine will automatically detect the length of your product, and then it will give you a graph. So therefore you can quickly screen so many let's say plants or like it's a fruit flies within a short period of time with the use of capillary electrophoresis over the conventional agarose gel electrophoresis. Okay, so probably like some of you will do the this you will do this. I mean, follow this approach um, when you do your directed study. But you see, this is going to take a lot of time. But unfortunately, we don't have a capillary electrophoresis unit or the fragment analyzer. There's one in the university, but it's not functional. It's there for like many years, but it's like a big white elephant that uh, no one can use, unfortunately, at science faculty. Okay, so moving forward, we talk about the gene markers, biochemical markers, and then we talk about the tandem repeats, short tandem repeats, or the SSLPs, and they are under that two different ones, mini satellites and microsatellites. And if you think about all these ones, so I'm pretty sure some of you have an idea of like having even much better marker, still a DNA based marker, because we are talking about like initially big genes, then like some biochemical parameters, and then short time derivative. Can we go even smaller? Of course we can. So we can use single nucleotide polymorphism, and nowadays we consider SNPs or SNPs as the most useful type of DNA markers you can find. Okay, so and if I'm pretty sure you already know what is a SNP. A SNP is basically a position in your genome. Okay, so if you take like let's say like one small fragment of your genome, so there can be variation like single nucleotide. Maybe one of you have G and maybe I have C. Okay, so that means there's a difference. So these ones are we call as SNPs. And if you look here, you can see, so here there are like if you look at allele 1 and allele 2, those who contains allele 1, so you have G, and then the allele 2 you have C. Okay, and if you think about the species, there are like a large number of SNPs you find in your genome. If you think about human genome, so when they did I think the thousand genome project, they sequenced more than thousand. So they looked at around 2,500 genomes, and what they discovered was there are 84 million SNPs between these 2054 human humans. Okay, those who provided their genome. So like let's say like if you compare yourself with like other members in your class, you will see there's like millions of single nucleotide polymorphism. So therefore, so we are not similar, although we are like 99.9% similar, still we are different. Like every time a kid is born, so they are going to be a genetically modified organism, you like it or not. So all of us are like genetically modified because we are different. We are genetically modified compared to our parents. Okay. So let's see what is happening. Yeah. And if you take the whole human population, the whole civilization, so we estimate there are around 3 to 4. Six is for million, right? Seven is for billion, and eight is for I don't know, whatever, like the tens of billions. Like 3.3 to 4 into like a 10 to the power of 8 SNPs are expected among the human population. So that is the number. So that means, so you have a golden opportunity to develop like pretty cool SNP markers in order to do your genetic mapping. Okay, so now probably you're thinking, okay, so we already discussed about the RFLPs. Okay, so if there is a change like this, why not we have like let's use a restriction enzyme. So one probably cut this one, other one is not going to cut. Okay, but if you think about reality, it is not the case, right? Why? Because uh, so there are limited number of restriction enzymes. Okay, so there are some allelic variations. They can't be detected by any restriction enzyme available for us. Okay, so there are around, I think, 6,000 restriction enzymes available for us. Still, we can't cover this the multi-billion SNPs found in our genomes. Okay, and so if you think about the SNPs, so I'm pretty sure you're thinking, okay, so now can have, if you have A in one allele, so the other allele, there are four possibilities, right? A, C, T, G. Okay, so that means so they are like in a multi-allelic, but if you think about the reality, it's different because if you take the SNPs, vast majority of SNPs are biallelic. Okay, because even though we expect one of four nucleotides to be there in our specific allele, okay, but it is not going to happen practically because theoretically, of course, it's possible, but in real practice, most SNPs are just two variants or they're biallelic. Okay, why? Because a SNP will originate when a point mutation occurs in, let's say, your parent's genome, like one. Okay, and there, what is going to happen is like one nucleotide is basically converted into another. Okay, just one. And so then if this happens in somatic cell, it's not going to do anything for the upcoming generations, but it will be stay with you, but not for the others. But if this happens in a reproductive cell, what is going to happen is one of the offsprings of that mother or father is going to inherit that mutation. And if this mutation is not lethal, what is going to happen now? If that person is going to produce more offsprings, so that way, so this allele will basically spread across the population, and then it will become established in the population, that specific population. Okay, so now you have the original, the sequence plus the new sequence, just one. Okay, so the parental allele and then the whatever the allele found in progeny. Okay, so therefore there are only two alleles. So in simple terms, it's the original sequence and mutated version. And if you want a third allele to occur, what is going to happen? So now another mutation should happen in that exact same location, and that should replace so one of the nucleotides it had earlier. Okay, so then so now it needs to go through the same process. Like you need to make like a many children over the generations, and then only it's going to get established in the population. Okay, so therefore this scenario is not impossible but highly unlikely. Okay, so therefore, most of the SNPs are going to be biallelic. Okay, now you're thinking, okay, so these SNPs they have only like two forms, so therefore it's less use for us as markers. But in reality, don't forget like earlier slide I mentioned there are billions or more than billion SNPs. So therefore, this disadvantage of being biallelic is basically mirrored or the the term eliminate not eliminated but nearly eliminated by the presence of many SNPs. If you take most eukaryotes, they have at least one SNPs in every 100 base pairs of DNA. Okay, so therefore the SNPs will enable us to make a very detailed genome maps. Okay, and next we are going to look at how we can type these SNPs, and there are different methods, very cool methods you will see. And because this the the era of SNP markers came very late, so therefore we have a lot of new techniques to screen SNPs in very large scale or high throughput. Okay, so most of the methods they are based on this what we call as oligonucleotide hybridization analysis. what is going to happen there is, give me 1 second. Zoom is, I have to log in again because somehow uh, the it automatically got signed out. Give me 1 second. Let me quickly log in. All right, we're back. Okay, so we were talking about the oligonucleotide hybridization, and I'm pretty sure you're like very familiar with oligonucleotide. So every time you order a primer, so that is an oligonucleotide. Basically, an oligonucleotide is like a small fragment of single-stranded DNA molecule, and usually it's less than 50 nucleotides, and they are synthesized inside a tube, or like it's like an in vitro or like synthetic compound, a synthetic oligonucleotide, so that you can use for your advantage. What is going to happen is this oligonucleotide, because it is single stranded, uh, when there's a right combination, it will hybridize with another DNA molecule, and then it's going to make a perfect, a completely base paired structure with the second molecule we are like basically adding. If I show you in like more visual manner, you can see what happens is you have your oligonucleotide that is synthetically basically we uh, fix it to a slide or array, and then it is like that. Let's assume it's located in this way. So now you basically you put your DNA fragments. So then if there's a sequence similarity, they are going to hybridize. So the cool, interesting thing is, let's say there's a mismatch in the middle. So then what is going to happen is, if you provide the correct condition, it's not going to basically hybridize. So therefore, there is no match, or it's a mismatch. So therefore, you are not going to see any molecule hybridize. Okay, so we can use, what we do is basically we use fluorescent pigments in order to detect this hybridization or lack of hybridization. So then we can visualize it like this. So this is basically a SNP microarray. So there are sometimes like 40,000, 50,000 different oligos showing this individual or the SNPs quoted, or using a robot into the slide. And when we basically fraction our organism DNA, and then we look at the pattern or the fluorescent signal for the presence of that specific SNP. Okay, so this is a very simple and quick method. So by doing this, you can screen more than 50,000 SNPs using just one slide. So this is how they do most of the GWAS, genome-wide association analysis in like especially humans. You will see, I mean, they use these microarrays, and then one shot, you can screen all your favorite SNPs in one array. Okay. And also you can use other techniques. This is what we call as dye quenching methods. So what we do is basically we have a molecular beacon, and so this beacon, probably you will learn this one during because I know you have a course, something called in vitro techniques or something like that. So there, most probably you will learn about this. This is a lamp reaction. So basically, I think you heard about lamp when we talk about because remember good old days. When we had this the serious time of our COVID pandemic, we didn't have any test kits or the antigen kits. So those days, remember, one the Peradeniya lab, and it was already there in the other countries. So they came up with this lamp primer kit. So where basically you take a swab, and then you put it into a solution, and then you take that solution, and then So you put into like the second the to the specific the special tube, and then when you basically heat it, it will give you a color. So that will give you an idea whether you are positive for COVID or not. And so there are different ways to visualize it. So here you can use the same technique in order to visualize the presence or the absence of SNP. Okay, so this is what we call as dye quenching methods. Here you have the microarrays. Those are the two kind of popular methods people use in order to identify different SNP markers. And there are a couple of other types. If you look at this diagram, uh, so there's another one called the oligonucleotide ligation assay. There, what is going to happen? So you basically have a kind of a foundation, and then what is going to happen is if your SNP is there, so there's no mismatch, so they are going to basically bind, and then as a result, the ligation is going to happen. And if there's a no if there's a mismatch, the ligation is not going to occur. So then you can basically detect that. And then also you have this arms test, which the long term is amplification refractory mutation system. And there also you're using this mismatch in different here. So if there's no mismatch, it's going to basically create a PCR product that you can detect. If there is a SNP, there's no match, so that means not going to amplify it. So then you can separate the presence or the absence of specific PCR product. Okay, so these are the different types people use for the as markers for the genetic mapping. And we already discussed about maybe 5 or 6 different markers people use in order to uh, do the genetic mapping. Okay, so that's all I'm going to talk about the genetic maps. But you have to have some kind of a basis to do the mapping part, right? So there, I will give you like a very brief overview, and I want you all to read about it as a homework. But before that, I think I'm pretty sure I just want to give you like kind of a refresher. So what is the when we talk about the basis of genetic mapping, you can't forget about the Mendel. So remember, so you have purple parent and then the white parent. So when you cross them, so they will basically give different type compared to self-fertilization. Because if you self-fertilize purple one, it's going to create a purple one. So the violet color one, and then so likewise, like in each generation, it's going to create a violet color. And if it's white, so you can see it's going to create white because there's no mixing. If the white one is VV, assuming this is diploid, the white is WW. Now, if you cross violet flowers with white flowers, what is going to happen is whatever the dominant trait is going to appear, although it's heterozygous. So this one is homozygous. VW is heterozygous. That is something you should know. And then this is the ideal scenario when you learn about Mendel's laws, you learn about this. But in reality, there are a lot of different scenarios, possible scenarios. To give you like two example, one is like incomplete dominance. So what is going to happen is it's going to give like an intermediate phenotype compared to the original, the paternal phenotype. And then some can be this, sometimes there can be codominant, so that means they express both alleles. So therefore, you know so you have to think about all these different possibilities. And so I want you to read, and there's a small video associated with this link. So go ahead and read this, and then watch the video. So this should give you a very brief overview about genetic mapping and how it is done. Okay, and moving forward, so we talk about what kind of genetic mapping we can do, and then what are the markers we can use. And next, we are going to talk about the limitations when it comes to genetic mapping. We already discussed about some limitation, but we are going to uh, discuss a few additional limitations as well. And So your the resolution of your genetic map is going to depend on the number of crossovers that have been scored. Okay, because if there's no variation, there's no way you can do the genetic mapping. Okay, and this is not a big issue for microorganisms, because remember, if you take E. coli or Escherichia coli, so they can multiply within 20 minutes, but it's not going to be the same case for the others. Okay, and if you can multiply them within 20 minutes, so that means you can obtain large numbers, and then that enable us to, have many crossovers to be studied. Okay, so that means, so you can have very highly detailed genetic map with the help of the prokaryotes. And if you take an example, when the Escherichia coli, the E. coli genome sequencing project started, it was in 1990, the latest genetic map at that time, it comprised 1,400 markers. So that means an average of one marker per 3.3 kb. That's pretty good, right? Because this is sufficiently detailed to ensure the mapping, uh, the genome sequence they are getting to the genome assembly. Okay, and so that's why, working with prokaryotes, sometimes there are a lot of advantages. And similarly, if you think about the second sequence genome, the Saccharomyces cerevisiae, and this is also supported by what we call as fine scale genetic map. So that means very comprehensive sorry, genetic map. So they had approximately 1,150 markers. So that is on average one marker per 10 kb. Okay, so therefore it was not difficult with these small organisms, or like a unicellular, unicellular organisms with small genomes and prokaryotes. But the issue is again with humans and most other eukaryotes, because they have uh, of it's kind of impossible to have large number of progenies within a short period of time. So, and like in humans, I told you, right? You can't basically get 100 people and then 100 males and 100 females, and tell them, okay, go and cross over. We are going to look at your children. So that is not a reality. And same for the other most of the other eukaryotes as well. So therefore, we can study less meiosis events compared to the others. So therefore, the resolving power of our linkage analysis is very restricted. So what this means in simple terms? So that means whatever the markers we are getting as they are several tens of kilobases apart. And when you look at your map, so now you see they are located same portion in your genetic map. So that will reduce the accuracy. Okay, so therefore the our genetic maps, they have a limited accuracy, because the good old days, people assume that crossovers occur at random along the chromosomes, and this is not 100% true, because I'm pretty sure you heard this term hotspots. Because if you take your chromosomes, so they have what we call as recombination hotspots. Okay, so that means some places are more vulnerable for mutations or crossovers compared to the other positions. Okay, and this was very evidently displayed when the genetic map of Saccharomyces cerevisiae, or the brewer yeast, was published, because so their chromosome 3, so there was an issue. Um, and don't forget that the it is the first eukaryotic chromosome to be sequenced, and it helped us to do a direct comparison between genetic map versus the positions of markers as shown by the DNA sequencing. And there were considerable discrepancies, and it was even if you think about the extents, that one pair of genes, they were like completely ordered incorrectly by the genetic analysis. Okay, and don't forget that the yeast was one of the two eukaryotes compared to the fruit, like the fruit fly, uh, that had a very good genetic map. Okay, so both had like as I mentioned earlier, very early. So they were like they had like very high resolution genetic map. Still, there were some errors, as you can see here. So you can see, so here you have your genetic map, and here you have their physical map. You can see in ideal scenario they are like this. Okay, like direct lines, no crossing. But when we compare it with the physical map, you can see. So initially, based on genetic map, you thought GK1 is located here, and CHA1 located right next to it. So this was the order. But after they came up with the physical map, they realized, okay, no, CHA1 is located first, followed by GK1. So and they had a huge gap. So you can see. So there was like huge discrepancy when it comes to the assembly. Okay, so now the question for you to ask yourself, if the yeast genetic map is so inaccurate, how precise it's going to be the organism with less genetic mapping? Okay, so therefore we need a different approach. So next week we are going to discuss about physical maps. So we will see how we can use physical maps to have a much better resolution and much higher accuracy. Okay, so that's all I'm going to talk today. And if you have any questions, I'm happy to answer.